Dear students, in this lecture I will discuss lipids as the respiratory substrates from the chapter Respiration of the course Plant Physiology 1. I am Dr. Smina Mahmood, Assistant Professor Botany in the University of Education, Lahore. This lecture is the continuity of the previous lecture in which I started lipids as the respiratory substrates. In that lecture, I introduce you what is lipid, what is the structure of lipid, and what is the functional importance of the lipids. In this lecture, I will tell you how lipids get synthesized in the body and how lipids get enter into the respiratory pathway. Because I told you respiration is our target to discuss in each and every lecture. We always discuss carbohydrates as the major source of energy and respiratory substrate in the body. However, lipids are present in less quantity in any body as compared to the carbohydrates, but lipids and carbohydrates in most of the instances they get metabolized side by side because when in a body required higher amount of energy while walking when you are walking fast or running or you have some muscular activity in that time you need more energy and carbohydrates alone are not enough to fulfill the requirement of the body, so lipids get metabolized although carbohydrates are already metabolizing in the body. And it is also the fact that lipids metabolize less, but the energy rate or rate of energy which is produced after lipids metabolism is more as compared to the carbohydrates. As this picture represents, which is the example of Arabidopsis leaf, and it shows the distribution of dry weight as the percent of total, and dry weight is approximately 8% of the fresh weight. Here in this you can see that among all the components which are present in the leaf, carbohydrates contains 59% of the total. Rest is the 19% is the protein, 9% are the mineral, 7% any other, and 6.1% are lipid. Here you can see the big difference between the quantity or percentage of both biomolecules in the dry weight or in their distribution. The lipids are 6.1% and carbohydrates are 5.9%. As I told you that carbohydrates are present in huge quantity and mostly utilized in the body. And if we further see the distribution of the lipids, here we can see that 3.2 of the 6.1% is the glycerolipids. 2% 0.2% is the waxes, 0.3% are the cutin monomers, and 0.7% are the others which may be composed of plastocoanin, carotenoids, tocophenols or sterols or any other, and chlorophyll is 1.5%, and sphingolipids are 0.2%. This is the another example which is the which shows the distribution of dry weight in the Arabidopsis seed. Here you can see that lipids are present of the total 37%, and carbohydrate quantity is 20% of the total. Proteins are 30% and 13% composed of other biomolecules. And among these 37%, 94% of these 37% of lipids are the storage lipids. Here it is very interesting that you see in the previous slide I show you the dry weight distribution of the leaf, and this is the dry weight distribution of the seed, and you compare that in the leaf carbohydrate quantities are more, that is 59%, than the lipids, which are 6.1%. As leaves are the photosynthetic organs in the plant body, so more the leaf photosynthesize, more carbohydrates are synthesized, so more is the quantity of carbohydrates in the leaf. But seeds are in the plants are considered as the storage house for of the excess food. And in the previous lecture, I also elaborated you that the extra amount or surplus carbohydrates, which are surplus than the needs of the plant, they get converted into the lipids and stored into the plant body. So seeds serve as the storage house. So due to this reason, the surplus food gets stored in the form of lipids. So the concentration of the lipids in the seed is higher as compared to the carbohydrates. And here you can see the clear difference, almost 1:2 ratio. As fatty acids as the structural component of lipids are very important, so it is important to discuss where they synthesize and from where they get exported to the other parts of the plant where they are not synthesized. So fatty acid synthesis differ from all other eukaryotes in two fundamental features. First, unlike the cytosolic location in other kingdoms, fatty acids are produced in the plastid compartments of the plant cell, and chloroplasts in the green tissues and proplastids or leucoplasts in non-green tissues. Secondly, the plant fatty acid synthase, which is the enzyme, is dissociable complex with separate proteins for the acyl carrier proteins and each enzyme. This biosynthetic pathway is not restricted to any specific tissue or organ, but it is found in every cell of the plant because chlorophyll is present in each and every cell of the green part of the plant where photosynthesis occurs. So where the plastids are present, where the chloroplast is present, over there fatty acid synthesis side by side to the photosynthesis of the carbohydrates is carried out in all the cells of the plants all the times. As you can see in this picture, this is the cytosol. This inner double membrane bounded is the plastid. This is endoplasm reticulum and different organelles which are present in the plant cell. So here you can see here is the carbohydrates, sucrose and other derivatives are present, which are under the process of glycolysis, which are present in the cytosol. However, from where pyruvate get entry into the plastids, and over there by the action of the enzyme and through acetyl CoA conversion, it get enter into this cycle where fatty acid synthase overall, as you learn the function of fatty acid synthase in the chapter of photosynthesis, and that how it function in the synthesis of the ATPs and in the synthesis of fatty acids. So with the help of this enzyme, you it get converted into the fat. This is fat. You can see here the structure, and when it gets synthesized over here, after synthesis, it get exported from this membrane to the other part of the cell, like here in the endoplasm reticulum. So it is synthesized in the plastids and it get exported to the other parts of the body or the cell where it is required. As we are more interested in the catabolism of fatty acids because in this process energy is released, and in this way it enters into converted into the simple compounds and get entered into the respiratory pathway. So fatty acids are the most important source of energy, especially in the absence of carbohydrates, which is released by their oxidation. Three types of fatty acids oxidation pathways are present in the cells, that is beta oxidation of fatty acids, alpha oxidation and the omega oxidation of the fatty acids. Among other types of oxidations, beta oxidation is most important, so I will discuss only beta oxidation in this lecture only. This beta oxidation is the process by which fatty acids in the form of acyl CoA molecule are broken down in mitochondria or in peroxisomes to generate acetyl CoA that can get enter into the Krebs cycle. And this breakdown occurs in the oil body of the oil storing seeds and in the peroxisomes. Here you can see in the figure that triacylglycerol molecule in the presence of lipase enzyme get breakdown into its components fatty acid and glycerol, and in this step glycerol is released into the cell, but fatty acid get into another round of metabolism. And this metabolism occur in the peroxisome where by the action of the enzyme ATP get utilized in this process and it is broken down into acetyl CoA, and this whole process is called the beta oxidation. Now I will discuss in detail how the whole procedure is divided into three main steps. The three steps of the oxidation are the activation of fatty acids in the cytosol, secondly, transport of the activated fatty acids into the mitochondria via carnitine shuttle, and third and the last step is the beta oxidation that occur in the mitochondrial matrix. So this is the first step, that the activation of fatty acids in the cytosol, and this activation occur in cytosol and the enzyme which is used, that is fatty acid thiokinase or acyl CoA synthetase, which is present in the cytosol. So in the presence of this enzyme, fatty acids by the using of ATP and this in the presence of this enzyme get converted into the activated form of fatty acids, and with the release of AMP molecule and two inorganic phosphate. So first the fatty acids get activated and converted into fatty acid acyl CoA, a long chain molecule. After conversion, this molecule get transported into the inner side of the mitochondria, and this transport or occur through an enzyme called carnitine, and this is the structure of carnitine. Overall here you can see, and you can see the charge and the components which are present in the carnitine. So as this is enzyme mainly facilitate the inside and outside movement of the fatty acid across the membrane of the mitochondria, so this is called carnitine shuttle. Here you can see fatty acid carnitine and fatty acid carnitine inside and outside the mitochondrial membrane, and this is the activated form of the fatty acid that get transported into the cell for further metabolism. This is an other picture that shows that outside and inside boundaries of the cell, and fatty acid first present outside the body, and with the help of transporters it get transported into the inside of the cell, and after getting into here it get activated, and in the presence of acyl carnitine get entry into the mitochondrial membrane where it get metabolized, and the metabolic products at the end, that is the acetyl CoA, get enter into the Krebs cycle, which is the major energy producing step of the process of respiration. This is the final step of the beta oxidation of the fatty acids, and I show you in the last picture the first step after which acetyl CoA is produced, that is the substrate for the Krebs cycle, and directly utilized to produce ATPs in the process of respiration. So this final step is further divided into four steps. First is the oxidation by FAD-linked dehydrogenase enzyme. Secondly, after dehydrogenation, hydration by hydratase enzyme occurred, and after this hydration, again another round of oxidation occurred by NAD-linked dehydrogenase. In first oxidation, FAD-linked dehydrogenase is used, while in the second oxidation, NAD-linked dehydrogenase is used. And in the last step, thiolytic cleavage biothiolase enzyme is occurred in the cell, and after this the fatty acid is get converted into the acetyl CoA. That is the final product of the beta oxidation of the lipids. Now it is interesting to see that all the processes or three steps I told you, and the further subdivision of each major step, that is under the control or it is catalyzed by the use of enzymes. Here you can see dehydrogenase, hydratase, again dehydrogenase and thiolase. So enzymes play a very important role in the activation and deactivation of any kind of metabolic or anabolic reaction in the body. This is the schematic representation of the third and final step of the beta oxidation process. Here you can see this is the first one in every step is showing the substrate on which this represents the enzyme. This enzyme act on this molecule to get it converted to this form. So here acyl CoA, in the presence of acyl CoA, dehydrogenase get converted into the trans-enol CoA, and again this product of the first reaction act as a raw material for the next step. So this trans enoyl CoA is get converted by the use of enoyl CoA hydratase. As I told you in the last step, first dehydrogenation occur by the use of FAD linked in dehydrogenase enzyme. And here this is the hydration process. So trans enoyl CoA get hydrated in the presence of enoyl CoA hydratase and converted into the beta hydroxyacyl CoA. And in the next turn, beta hydroxyacyl CoA in the presence of NAD linked dehydrogenase, which is called beta hydroxyacyl CoA dehydrogenase, get converted into the beta ketoacyl CoA. And this beta ketoacyl CoA, after the action of thiolase enzyme, which is called acyl CoA acetyl transferases, also get converted into the product acyl CoA plus acetyl CoA, and this acetyl CoA get enter into the Krebs cycle into the mitochondria, that also occur into the mitochondria. This beta oxidation, the final round of the beta oxidation occur into the mitochondrial matrix, and also in the mitochondrial matrix process of transpiration, and especially the Krebs cycle and electron transport chain. These two processes operate into the mitochondrial matrix. This is the summary of the whole process of fatty acid synthesis and finally its metabolism to release energy. So here you can see fatty acid gets synthesized into the plasmid. This is double membrane bounded organelles. Fatty acid may be synthesized in the process of carbohydrate synthesis, in which extra or surplus carbohydrates get converted into the storage form of fatty acids, or maybe lipids synthesized in the outside of the plastids. They get transported by the other organelles into the plastids. So from here, by any means, it is synthesized. Fatty acid get converted and exported to the other part of the body. From there, it is by the use of carnitine shuttle, it get transferred to the mitochondria, where beta oxidation of the fatty acids occur. That further operate into three steps. And in the last, the product of the beta oxidation is acetyl CoA, and that acetyl CoA is no need to be further exported outside of the mitochondrial membrane, because the Krebs cycle and electron transport procedure of the respiration process also occur into the mitochondria. So as both processes are occurring in the same place, so the product of metabolic process get utilized into as a substrate in the next metabolic process, that is called respiration. And in response of that utilization in the process of respiration, ATPs, which are the currency of energy in the cell, are released. As I told you earlier that both fatty acids and carbohydrates used as a source of energy in the body side by side. And I also told you, no doubt, carbohydrates are used more, but the rate of energy which is produced as compared to the fatty acids is less. Fatty acids are need in more energy to get oxidized, but small amount of fatty acids produce more number of ATPs. So in the condition when high energy is required by the body, in that situation, fatty acids maybe in the absence of carbohydrates or maybe in the presence of carbohydrates, side by side they get metabolized into the ATP to produce ATP. So here in this equation you can see if 180 gram per mole of carbohydrate used, and in the process of glycolysis and all of this, here the detail you can see of the each step, which enzyme is used, and what amount of ATPs are utilized or produced. So the final product which is important to know for us, that this 180 gram per mole produce 30 to 32 moles of ATP in one reaction, while as compared to this molecular weight of 256 gram per mole of one fatty acid molecule, and this produce ultimately 106, that is 106 moles of ATP, and both are showing ATPs per gram. This is ATP per gram, and this is also the ATPs of 1 gram. So you can see how important fatty acids are important into the body, and why they are required in the process of respiration. I hope in the end you all will be able to understand how fatty acids get synthesized and how they get metabolized into the body to release energy, and how the end product of the metabolic process of fatty acids get utilized or get entered into the respiratory pathway, which is our main concern. I use these two books to prepare this whole lecture. If you are interested, you can consult with these books. Thank you. Thank you.